Vanakkam Makkale. Today we'll be seeing about the mechanism of HCl secretion. So before diving into the topic of mechanism of HCl secretion, let's see about why do we need HCl in the first place. Out of the many functions of the stomach, secretion of HCl constitute one of them, right? So why do we need HCl? There are four main things. HCl aids in the digestion of the food. It kills microorganisms. It converts Fe3 plus to Fe2 plus. That is ferric form of iron to ferrous form of iron. So why do we need the conversion of Fe3 plus to Fe2 plus? Is because Fe2 plus is only the absorbable form of iron. So HCl வந்து conversion of Fe3 plus to Fe2 plus வேணும்னு சொல்லியிருக்கேன், right? So when there is deficiency of HCl, Fe3 plus is not converted to Fe2 plus. What happens? There is deficiency of iron. So what will develop iron deficiency? Anemia. The last function of HCl is conversion of pepsinogen into pepsin. Pepsin is an active product. It breaks down complex protein molecules into smaller protein molecules. On to the mechanism of HCl secretion, right? So what do we have here? This is a parietal cell from where the HCl secretion occurs. This is the apical membrane and this is the basolateral membrane. Parietal cells are present in the fundus and the body of the stomach. Apical side of the parietal cell, we have what is known as H plus and K plus ATPase pump. Why do we need an active mechanism for this? Because we are pumping H plus against the concentration gradient. Gastric lumen already there is more amount of H plus. Again we have to pump more H plus from the cytoplasm of parietal cell into gastric lumen. We will need to exert some energy. And this exertion comes by the hydrolysis of ATP. H plus goes into the gastric lumen and for that K plus comes from the gastric lumen into the parietal cell. So where do we get this H plus from? We get this H plus from the breakdown of H2CO3. Carbonic acid breakdown la H plus atom varudhu. Where do we get this carbonic acid? Carbonic acid is formed from the joining of carbon dioxide and H2O in the presence of an enzyme called as carbonic anhydrase. So carbon dioxide and H2O join achina in the presence of carbonic anhydrase enzyme, we get what is known as carbonic acid. In the carbonic anhydrase enzyme block pandradhukku, we have what is known as acetozolamide, right? Combination of carbon dioxide and water in the presence of carbonic anhydrase gives you carbonic acid. Carbon dioxide engindu varum? From metabolisms and the plasma we get carbon dioxide. So carbonic acid breaks into H plus ion and HCO3 minus ion. Carbonic acid gives bicarbonate. In the bicarbonate vandu from the cytoplasm of parietal cell gets into the interstitial fluid. Adukku badala we get chlorine, right? So chlorine from the cytoplasm gets into the gastric lumen, combines with H plus and gives HCl. Next thing is reutilization of the potassium ions. Potassium ions active pump மூலமா it is getting inside the cytoplasm of parietal cells. இங்கேயும் பார்த்தீங்கன்னா from the interstitial fluid potassium ions are getting inside the cytoplasm for exchange of the sodium ion. So there is more amount of potassium in the cytoplasm which by passive diffusion gets into the gastric lumen, maintaining the adequacy of K plus ions. இங்க வந்து K plus ion adequate இருந்தாதான் H plus ions can be pumped out with the help of H plus K plus ATPase pump. So this is all about the mechanism of HCl secretion. Carbon dioxide and water in the presence of carbonic anhydrase enzyme gives you carbonic acid. Carbonic acid வந்து breaks down into bicarbonate and H plus atoms. Bicarbonate exchanges with Cl minus. H plus ions is transferred into the gastric lumen by the H plus K plus ATPase which joins with the chlorine ions forming the hydrogen chloride. So after seeing about the mechanism of HCl secretion, we'll be seeing how the secretion is regulated or the factors that influence the secretion of HCl. The factors that are given above increases secretion of HCl and the factors that are given on the side decreases the secretion. So the first one is acetylcholine. Acetylcholine is produced from the vagal cholinergic fibers. This acetylcholine acts on the M3 receptors which is present in the parietal cell. Upon acting on the M3 receptors, intracytoplasmic concentration of calcium ions increases. This in turn regulates the protein kinases, increasing the HCl secretion. So acetylcholine not only acts on the M3 receptors but also acts on the G cells as well as the enterochromaffin-like cells. Acting on the G cells stimulates the production of gastrin. Acting on enterochromaffin-like cells stimulates the production of histamine. So G cells produce gastrin which acts on the G receptors present on the parietal cell, increasing the intracytoplasmic concentration of calcium ions, thereby increasing the secretion of HCl. Histamine acts differently, right? It acts on the H2 receptors present in the parietal cells which increases the cyclic AMP. This cyclic AMP regulates protein kinases and increases the secretion of HCl. So the three factors that increase the secretion of HCl: acetylcholine, gastrin, and histamine. Distension of stomach can also increase the secretion of HCl. Now for the factors that inhibit the secretion of HCl, we have somatostatin, secretin, and the autoregulation methods. Somatostatin decreases cyclic AMP, thereby decreasing the HCl production. Secretin is produced when the chyme enters the duodenum. So what is chyme? Chyme is partially digested food. This partially digested food, when it enters the duodenum, it produces secretin. Secretin decreases the production of HCl. So what is this autoregulation of HCl? When HCl concentration in the gastric lumen increases, the pH of the stomach drops, right? When the pH drops below 2, there's a process called autoregulation of acid secretion. By negative feedback mechanisms, HCl secretion decreases. That's all about the mechanism and regulation of HCl secretion. Thank you so much.